Thank you for the introduction. Thank you, Dr. Kali Selvi, for giving us an hour's time where we learned much more about CSIR. I have known CSIR for a long time, but she explained to me all that's being done at CSIR, the technologies you have developed. Congratulations, madam. And with your dynamism, I'm sure things will happen. Much more things will happen. Many more things will happen. I'll briefly tell you about uh, 60 years of science at IISc. I joined the Department of Biochemistry as a PhD student in 1961, and my guru was Dr. P.S. Sharma, Padmanabhan Subbaraya Sharma from South Canada. I started actually with this dal called Kesari dal. And one really doesn't know the tribes in central India take up, consume this dal. And I do not know whether it's still correlated with leg paralysis or they are not able to walk. Even today, it's not very clear. But we took up isolating a neurotoxin from this dal, and we isolated a toxic principle, and we characterized it as oxalyl diaminopropionate acid. But the cause of neurolaterism, as it is called, is still not known. Although this is a neurotoxin, it will produce cerebral malaria in the animal model and so on. But whether it is responsible for the human disease, we do not know. Well, of course, I worked with Neurospora. I don't want to spend much more time. But the only issue I think is I started with a CSIR project with one JRF and 5,000 rupees consumable per year. I'm talking about 1960s. Probably means 6 crores now, probably. Characterize this molecule called desferrioxamine. It's a great iron binder. That's why I got involved in iron metabolism. And interestingly, with high affinity for iron, it's still able to donate iron for heme synthesis. Therefore, desferrioxamine is used for treatment of thalassemia. I'll skip this for want of time. So, iron is donated. There's a pathway of heme biosynthetic pathway. I will not get into great detail. And the last step is called as ferrochelatase, and it gives rise to this molecule heme, which contains iron, which is incorporated into protoporphyrin. And we worked with the fungus at that point of time. Then Satish Dhawan became director of the Indian Institute of Science. And I was spent 3 years after my PhD, didn't want to go abroad. And maybe I had a wrong impression that everything can be done in India. Why should I go abroad? I can do everything in India. That was the kind of thinking I had. And people said, you will not get a job in IISc without a tag, US tag. But interestingly, Satish Dhawan became director. And he said, if this guy has done so well in India, why are you looking for somebody from outside the country? Appoint him as assistant professor. It was Satish Dhawan who made me uh, assistant professor. And then in 1973, as a Fulbright scholar, I went to University of Chicago. There you see Myra Rabinovich, my mentor, paralyzed hands, legs, but still coming to the lab 5 days a week. So then I felt, if this man with this kind of inability could spend 5 days a week with all faculties intact, we should put more effort. That was, so I went 10 times to his laboratory in the University of Chicago. Murray's lab 10 times between 73 and 86. And that is where I picked up techniques, gene cloning, sequencing, nucleic acid hybridization, all the modern molecular biology techniques I picked up in Murray's lab. Well, when I came to India, I thought I should transfer this to animal system. It's all right, neurospora, fungus. So I started working with liver cytochrome P450. Cytochrome P450, as some of you would at least know, plays an important role in drug metabolism. Whole family of enzymes in the liver which are responsible for the metabolism of all kinds of drugs. In fact, almost every drug that is metabolized, which we take, is metabolized in the liver, and cytochrome P450 must be playing a role. It makes the hydroxylase, it adds a hydroxyl group. So I standardized several molecular biology techniques in the lab. I know how to quantify mRNA, how to run on nuclei, transcription in nuclei, promoter GFIC. I'll not get into all these, probably not needed to go into all these technologies in great detail. But we were the first to do DNA sequencing in India. At those days, I sequenced some 300 bases and went to the whole world. Oh, we have sequenced your DNA 300 bases. Today, probably 3 billion base pairs are sequenced within a week. That's how technology has moved, actually. Of course, it's, those days, because PCR was not known, so we had to quantify mRNA by translation. If we have more protein product is made, then more messenger RNA is made. That's all. Then we propose a model for how heme regulates cytochrome P450, classical methodologies which involve negative element, positive element, all this. We published proceedings of the National Academy of Sciences, USA. And we were very proud. We were the first group to publish some indigenous, without a broad collaboration, that we could publish in proceedings of the National Academy of Sciences from India. So we were very excited at that point of time. Once we identified this gene, we have a gene delivery. We thought this upstream, as we call it, sequence, can it be used because for gene delivery, for therapeutic purposes. Can you use a gene delivery approach into that? That was published papers we published. Anyway, the final conclusion of all the study in heme regulates translation of protein synthesis. Heme regulates translocation of proteins across membrane. Heme regulates transcription of P450. Heme is required for differentiation of hematopoietic cells. Heme is a multifunctional regulator. We could publish in Trends in Biochemical Sciences. That was a very important journal. At that point of time, AstraZeneca came to India. It was known as Astra Research Center at that time. For some reason, they came to me and sought my help to set up a lab in Bangalore. And, you know, I suggested 4 projects to them, one on tuberculosis, one on malaria, one on diarrheal diseases, one on cysticercosis, brain disease. Then it struck me, why I'm suggesting all this? Why should I not be doing myself at least one of the diseases? That's how I started malaria research. And, I don't want to get into great detail. For people who know about malaria, this is a basic diagram. It's all people who talk about malaria will use. The mosquito injects sporozoites, goes to the liver, quickly release, and then goes through a cycle, blood cycle. And then when the mosquito bites, it goes back and again develops into sporozoites, and the cycle will continue. And of course, curable with if it is susceptible to drugs, but it can also become resistant. It can also become resistant to drugs, malaria, placental malaria, for pregnancy, malaria is a big issue. But sometimes, placental malaria resistance develops is a huge issue actually. The question we asked at that time was, the parasite is in a sea of hemoglobin. When the parasite you culture, whether it's in vivo or in vitro, it's in a lot of in red cell, surrounded by a lot of hemoglobin. That means it gets a lot of heme from hemoglobin. Then why does it make heme? The biosynthetic pathway, the malaria parasite also makes heme by a synthetic route. We never understood why should it make heme? It is getting tons of heme from hemoglobin. Of course, pathway was worked by my students, published in Biochemical Journal, Journal of Biological Chemistry. Well, at those times, these were all big journals to publish. And then, of course, we have huge lot of papers. That's by a single student who worked out the entire pathway of how heme biosynthesis takes place in the malaria parasite. The heme dynamics in the malaria parasite is very interesting. It has RBC, reticulocyte. The point I'm trying to make here is, why does the parasite have so many routes to make heme? It gets heme from hemoglobin, it makes heme, and then the red cell makes heme, or reticulocyte makes heme. Why does it have to make so many? Why does the parasite have so many routes to acquire heme while it is getting tons of heme from hemoglobin? That was the question. Classical techniques, as I said, we were the first to introduce knockout the first gene, knock out the last gene of the pathway so that the parasite doesn't make heme. If it doesn't make heme, what are the consequences? These are kind of questions. But interestingly, we found if the parasite doesn't make heme, it doesn't grow in the mosquito. You can see when the mosquito takes up the parasite, is loaded with the salivary gland. But when you have a knockout, then it will not make. So this was a very important kind of finding at that point of time. Got widely quoted by the global community that about the findings which this Indian group has made. Why do the parasite make heme in the blood stage? It cannot be waiting for the mosquito stage. Why does it? Biosynthetic heme is not required for parasite growth in the blood stage, but is required to sustain its virulence. If the parasite doesn't make heme, it doesn't become virulent. That means if the parasite doesn't make heme, The parasite, the mosquito may have the parasite, but it can bite, but it can't produce malaria. So we have a mouse model, an animal model, we have a cerebral malaria model, we have anemia model. That means here in the Swiss mice, parasite infects and parasite grows and they'll all die at some point of time. In the C57 black, you will see the parasite produces cerebral malaria. Cerebral malaria is not produced in the this model of anemia model, but only in this C57 black. So it depends on genetics of the animal. Functional role of parasite biosynthetic heme. While hemozoin is an inert molecule, so far as the parasite is concerned, it is considered as pathogen danger associated molecular pattern. That means how does the parasite detoxify heme? Now we have human or animal have biliverdin reductase, bilirubin reductase. Heme is converted into bilirubin and is secreted in the urine. But parasite doesn't have. So it converts the heme into a polymer called hemozoin. So that is so we have few things like food vacuole, pH, unsaturated fatty acid in the membrane, proton ATPase. I'll skip some of these slides for want of time. You can see parasites in the brain. These are parasites that are tagged with a fluorescent label. So you can see if they are present in the brain, yes, you can detect it. But if the parasite cannot make heme, you don't see the parasite in the brain. So for the parasite to go into the brain, you really need, this. For example, proton ATPase. This is the wild type. This is the ferrochelatase knockout. I told you this is the last step in the heme biosynthetic pathway. If the gene is knocked out, heme is not made, and then proton ATPase is not made. Then the other step is you can clearly see conversion of saturated fatty acid to unsaturated fatty acid, stearic acid to oleic acid. That's not made in the heme biosynthetic deficiency. So again, all this are summarized in this particular slide where the heme dynamics anyway is again recapitulated for you. And we can reverse the effect. I told you if there is a knockout, heme is not made. But if the gene is added, now again heme is made. That's about it. This slide says. So we can show it with the luciferase tag. When it is tagged, you can now see when the heme is made, parasite is in the brain. So the interesting feature I want to point out is hemoglobin heme and biosynthetic heme. The parasite is living in a sea of hemoglobin. But then, it also makes heme. But it's a tiny bit compared to the heme it is derived from hemoglobin. So why does it make this heme? That was a question that has haunted us almost for maybe more than a decade. Why does it make heme? It is getting tons of heme from hemoglobin. Anyway, I told you, if you inhibit this heme, parasite is not able to grow in the mosquito. It's not become virulent. It can't produce cerebral malaria. Then we asked this question, can I use an inhibitor and develop a new drug? If I inhibit heme synthesis, will the will it become a new drug target to treat cerebral malaria? That was the question they were asking. And then many molecules were screened. Typically, that's how you do. You know the target, ferrochelated inhibition and inhibition of heme synthesis. Ferrochelated is the last step in the heme biosynthetic pathway. So after many screening, we found this molecule, Griseofulvin, can inhibit heme synthesis. Very interesting. The repurposed molecule. Why do you call it a repurposed molecule? Griseofulvin is extensively used to treat skin infection in children. But we found it inhibits heme synthesis, it also inhibits cerebral malaria. The advantage with this kind of molecule, you know everything about this molecule, about its toxicity, because it has already been used to treat children. Therefore, you don't have to worry. Otherwise, normally when you discover a drug, you have to do a preclinical phase 1 trial, phase 2 trial, phase 3 trial. Here we have a molecule which we know about everything, and we are only finding a new application. We are saying it can inhibit heme synthesis, so it can be used to treat cerebral malaria. So, Griseofulvin is an antifungal medication, but we also found it can be used to treat cerebral malaria. Anyway, I have given all that. What Griseofulvin does is very similar to what heme knockout. So, Griseofulvin is a very effective adjunct drug to artemisinin, derivative to treat cerebral malaria. We could publish it in Nature Communication in 2022. And normally, the complaint is scientists are very happy publishing in Nature and so on, and they don't take it for application. But we took it for application as well. We are collaborating with IISc, a private limited in Mumbai, and they are taking Griseofulvin to clinical trials. So because of COVID, it got delayed, but otherwise, hopefully, they will start the clinical trial with this Griseofulvin molecule. Now, the other drug I quickly want to cover is the curcumin. Curcumin from turmeric. Also, it works to treat cerebral malaria. Now, curcumin, I don't know, maybe 10,000 diseases have been treated with curcumin if you look at papers published. But it's not recognized as a drug for any application. And people also don't believe, curcumin from turmeric, it's at a short half-life. It cannot become a drug, especially the Western world dismisses this. So I took it up very seriously. No, this is something there. I want to take it up in great detail. So we took it up. These are all various targets for curcumin. Curcumin, as I said, more than 10,000 publications as of 2016 on curcumin from turmeric. And it must have been much more, but still not an approved allopathic drug for any ailment. I thought this is no way. We have to take it forward. Why curcumin is not an approved drug? It can cure almost any disease. You look at literature or our Vedic literature onwards, curcumin is known from turmeric. It can cure almost any disease. That's why turmeric is such a very important ingredient in your culinary efforts, whatever it is. But don't underplay it. The curcumin that is present in turmeric plays a very, very important role. But normally, it can cure almost any disease. It works on so many molecular targets. Its bioavailability is poor. It's not patentable. So people, will not take it forward. Yes, the Western world. arguing with all these people. They publish essential medicinal chemistry of curcumin. Curcumin is a feel-good factor, they said. It doesn't do anything. No, I didn't leave it. So an effective drug should have adequate concentration, half-life, and so on. I said, no, that's not the way curcumin works. Curcumin works as medicinal chemists always will think, what is its half-life? How stable is this molecule? I said, no, curcumin works in a different way. It is don't, dismissal of an entire area of research is like throwing the baby out of bathwater. It's beyond the well-accepted tenets of medicinal chemistry. So I told them, curcumin is a uh, immunomodulator. That's how it works. Curcumin modulates immunity in the body. That's why turmeric is so important in the diet, actually. And many experiments I've done. Only one experiment I'll try to explain to you. Is the animal model. When infected with the parasite, suboptimal concentration, you will see the parasite slowly appearing, depending on the concentration you are using. At this concentration, it will die only after 34 days. But if you added 3 oral doses of curcumin, not a single parasite will come, not a single animal will die. Therefore, it's a very powerful anti-malaria. That's how, it's a very important experiment. I have done many experiments, but I thought this is the only one you want to highlight, actually. And we have done Western typical biochemists do. How you analyze the serum of the individual and you find anti-parasite antibodies in those who have taken curcumin. So that's how we say it's an immunomodulator. So we have cerebral malaria model, and we have, I don't want to again say a point which I am repeating with now, curcumin. To take care of criticism that curcumin uptake is poor, we could make nanocurcumin also. And nanocurcumin, you can clearly see with curcumin, if this is the clearance, this is the parasite, by tag. But if you have nanocurcumin completely clear, malaria in the brain, you don't see any parasite. So this is the model. How a cerebral malaria in the animal model is created at least. You have enough kappa B phosphorylation, you have breakdown of blood-brain barrier, and animals will die. But if you give curcumin, it will clear all this, but still anemia will be present and then a parasite will grow and animals will die. Therefore, if you have artemisinin, that is the standard drug for malaria, if you add curcumin, you will see not a single animal dies. Therefore, we have introduced a new adjunct molecule, the artemisinin, to treat cerebral malaria. Because one of the issues with cerebral malaria or malaria as such, even the adjunct drugs have become resistant. Therefore, we need an adjunct drug at least which will not become resistant. Hopefully, curcumin is a dietary molecule. Hopefully, no resistance will develop. That's how. So and of course, now many groups have worked. They have developed nanocurcumin. We have of course published all our papers. There are many other groups, including Western groups, have worked on this curcumin in big one. Therefore, we have a clinical trial, artesunate curcumin combination in uncomplicated and cerebral malaria. The National Institute of Malaria Research in Delhi is taking up the clinical trial. They said, the COVID has come in between, therefore they are delaying taking up this clinical trial. Of course, there's a hospital in Odisha that's also taking it up, and Institute of Life Sciences, Bhubaneswar, where I had a student who has become a leader himself, and he has taken it up in a big way. And I only correspond with them. They call me every week, Skype meeting and so on. So I am a sounding board at this point of time. So generally, you will see the Western world at least bias against curcumin and natural molecules. These things don't work. They are all feel-good factors. But it's very important for us to develop hardcore scientific evidence that these molecules really work. It's not based on Vedic literature, it's not based on old literature that we are saying all this. We have hardcore scientific evidence that they are working. So, many people use cell culture model. It's not adequate. We have to have in vivo models, and wherever possible, we have to use clinical trials. Of course, now curcumin combinations have 100 clinical trials for cancer therapy. Because I told you, curcumin has a wide application. It's an anti, NF-kappa B phosphorylation inhibitor, actually. The anti-inflammatory molecule. Therefore, it is a very wide application in cancer and so on. Of course, there's a company already which is tells me in Hyderabad, they have distributed 200,000 capsules of curcumin combination to treat COVID. And they said they have evidence that this combination works to treat COVID-19 at that point of time. Very recent clinical trial also has been done by this group. Oral curcumin and piperine is adjuvant therapy for the treatment of COVID-19. So what I'm trying to say is curcumin is a very widely can be used adjunct drug therapeutic purposes for a wide variety of. I do not know how our ancients knew such a molecule exists in turmeric and how it works. But but interestingly, there's all this much later we are able to find all this actually. So we have done molecular dynamics. I just want to tell you one thing. I don't know whether some of you may know the story of Madame Tu. She's a Chinese scientist. She discovered this artemisinin. It's a primary drug to treat malaria. Nobody ever recognized her. She did not even become a member of the fellow of the academy. This lady. So, then found artemisinin has saved millions, especially children against malaria. Finally, the US recognized her and gave her a Lasker Award, and then she got Nobel Prize in 2015. So I believe if she can get in artemisinin, there are so many contributors for the field of curcumin. Curcumin also should be of that category, in my opinion. Artemisinin has reached that category. Curcumin should be in that category. For me, it took a lifetime to identify two therapeutic molecules. One was curcumin, and I told you, Griseofulvin. Of course, this is basically the science I have done. Basically, two molecules, more or less, especially the malaria and cerebral malaria to treat. That is what. Of course, I have other involvements. Been chairing so many committees for DBT, DST, CSIR. You will name the organization I used to chair. In 2023, I told, I have done enough. No more committees for me. I said I'm quitting all the committees. But I'm still not able to implement it. But it's okay. Let me see how long it goes. I'm only 86 plus. So I've interacted with DBT right from its inception. And in 1982, it was started at the National Biotechnology Board. I was around at that point of time. So, we should start an organization for, this was part of DST at that point of time. Then, eventually all this I already mentioned to you. I used to work with CSIR earlier years closely with HRDG to conduct entrance examination. Eventually, that led to CSIR NET examination. Many of you would not know. Used to go to the Pusa campus, and we used to have, yeah. Also served as a member of the committee to select Bhatnagar awardees. No, those years. Now, I also been member of this chair of this, what is called as SPIRA, uh, uh, Biotechnology Industry Research Assistance Council, established by Dr. M.K. Ban, late M.K. Ban, who was Secretary of the Department of Biotechnology. He was the first ever to discover a vaccine in India. He made the rotaviral vaccine. Of course, I've chaired this BIRAC since its inception. As I told you, I stepped down from 14 committees last year. Now, BIRAC has been involved in discovering many molecules, many drugs, BIRAC's impact, is all statistics, technologies developed. I chaired the committee to develop indigenous vaccine against COVID. India made, which Western countries doubted in the beginning. India made DNA vaccine against COVID. India made messenger RNA vaccine against COVID. And I given the company, Zydus Cadila, made the DNA vaccine against COVID. Genova in Pune made the messenger RNA vaccine against COVID. Subunit protein vaccine was made by Biological Events in Hyderabad. And of course, vector-based nasal delivery was developed by Bharat Biotech. But this is where you need innovative thinking. I worked with so many messenger RNAs. I never thought they will ever become a vaccine in my wildest dream. But now mRNA vaccine has become a common parlance now. Everybody is talking about mRNA vaccine, messenger RNA vaccine. We also always used to think RNA is an unstable molecule. You can never do anything with RNA, only touch with DNA. Now this has become but this lady, Katy Kariko from Hungary, she had a very staunch belief in mRNA vaccine. She didn't even get a postdoc job in US when she migrated. But she was single-minded, intense. Now everybody is talking about mRNA vaccine. What next? The future of mRNA vaccine for every imaginable infectious disease. People are talking about mRNA vaccine. This is what Drew Weissman, is a well-known name in the field, is suggesting. Now, so far as CSIR is concerned, I have seen it has 5 verticals: digital and molecular surveillance, rapid and economic diagnosis, development of new repurposed drugs, personal protective equipment and hospital assistive devices, and supply chain and logistics. This is where, in my opinion, CSIR, this is taken from the website, actually. I just have to tell you, what has COVID, of course, we had a huge issue. It has taught us a lesson. COVID, vaccines normally take, or we used to believe they will take 10 years, to take 12 years. COVID vaccines were developed in 2 years. How is that possible? collaboration, international collaboration. The regulators worked with us to develop. That's how. That means the point I'm trying to make is it is possible to change, so-called dogmas. A drug will take 12 years, 15 years. The vaccine will take 10 years. Doesn't have to be. That is where we need innovative thinking. Can we develop it in less than 2 years? And this is where, uh, despite enormous development in modern science, of course, we still do not know how to handle this tiny COVID virus. I saw a report saying it's not a virus, it's a bacteria. Very recently, yesterday or day before yesterday. So, we still are not very clear uh, whether some diseases are due to viruses or bacteria, despite our, intense knowledge. So I believe we have to have a sense of humility and respect for nature, which will definitely serve the humankind, actually. Now, I I don't think I should have said a few suggestions, but anyway, that's how I wrote before I was talking to the DG. So government has come up with this Bio E3 policy. And of course, this is Bio E3 is what? Economics, ecology, and environment. These are the three E's. Food processing is one of the important components, and CSIR is in the area. My suggestion is, can we scale up biomanufacturing? I know CSIR can do manufacturing. Biomolecules, can it make? There are challenges in making biomolecules. Is it possible? Can we take up such a challenge? Probably it's already doing. I'm just saying it. Scale up biomanufacturing to replace classical methods that add carbon to the environment. Can we bring down the carbon we are adding to the environment? Biomanufacturing is within a fermenter. Hopefully, it will not add anything to the environment. And fast protein is an interesting concept. What do we biomanufacture? Can we biomanufacture microbiological proteins for food purposes? We only know of animal protein, we only know of plant proteins. Can microorganisms serve as food source? Food source, and can we, as a food component, is it possible? Can we manufacture those proteins? That will be a challenge. Because there will be a limitation if you are always dependent on animal and plant sources. We have to move to microbiological sources. If it is microbiological sources, then as a food component, it's an interesting concept. Can we promote manufacture of proteins from microbiological source as a food component? Can we do that? Is it bacteria? Is it fungus? What protein? Is it nutritionally savvy? This is where we have I believe CSIR is well equipped to handle digital technology. I initially wrote, but I know, it encompasses availability of open information on health, treatment, complications, and recent progress in biomedical research. But basically, I mean application of big data, artificial intelligence, telemedicine, blockchain platforms, smart devices in healthcare, and medical education. These are the areas which I thought, CSR should probably, is contributing already. My thinking, was, these are the areas which are of profound significance. And the most well-equipped organization, in my opinion, is CSR in this area. And, Internet of Things based medical devices known as smart medical devices. I'm sure you all know about this ability to measure glucose, oxygen, body temperature. Must be very, very useful. For example, a smart insulin pump can regulate insulin levels according to glucose readings, reducing the chances of both low and high blood sugar. A smart insulin pump can automatically inject adequate amount of insulin after measuring the glucose automatically. That's where, challenges. That's are the kind of things, in my opinion, which we should be doing actually. The in terms of figure, it will be like this. You have an insulin pump which will measure continuously the glucose in the blood and then inject the adequate amount of insulin. Is that possible? So this is my last slide. I just thought I'll give you a very brief. I don't know how relevant is this talk, but I thought I should share some of my thinking because I know CSIR has the sound technologies for some of the devices which I have been talking about and some of the areas which I have been talking about. Of course, I want to acknowledge our director, Dr. Govindan Rangarajan, because I told him last year, I'm already 86. I was made honorary, NASA honorary scientist. Should I take it up? He simply said, I look forward to your continued contribution to the nation and the institute. That means as long as you live, keep doing whatever you can. So I continue, and the group on malaria moved to, as I said, to Bhubaneswar, and he was my student. He is now the group leader, and they keep me active. I wrote two memoirs, one to reach the stars or dig the earth. Both are on doing science in India. Some people suggested, anyway, you are continuing, why don't you write the third memoir also? Third innings, I started, progress is very slow. But then I know there are things to write. COVID is not covered, Ram Janmabhoomi, International Day for Women Scientists. There are many topics I was mentally thinking, what is it I should write if I had to do third memoir? So, been involved in one way or the other in many of these things actually. I think I'm done. Thank you so much.
I'll just say thanks and then you can sit and also answer.
Okay, sure.
So, friends, I do not know to what extent you are all excited. I learned at least 1 dozen good points. Don't worry, I'm not going to list out all the 1 dozen points, but at least few points I really want to feel like sharing with all of you. See the kind of confidence with which Sir is talking. He's telling, I'm just 86 plus. Sir, definitely you have to live for many more years because Lasker and Nobel are chasing you, sir. Because I'm just taking the example that you showed, that is Madam Tu from China. So she was not initially recognized, but later on she got Lasker Award and Nobel Prize. And your adjunct molecule is closely traveling curcumin, and therefore curcumin should get Lasker, curcumin should get Nobel, meaning is you will get celebrated, sir. So days are not so far off. And sir, you openly made all of us to understand one more point. Your initial thought is valid even today. Should I really go to only other countries to really enrich my learning curve, to come back to India and to get a placement in IISc? You really created a record and you very humbly just appreciated the one, Satish Dhawanji. He was able to identify you as an assistant professor. And so you never disappointed the country's expectations on you. So country is even today feeling so proud that without even sending our people to other countries, by making our people to get themselves retained in India also, we can come up with a number of new discoveries in India. And sir, you humbly said that in your lifetime you have come up with two molecules. And your first molecule is a very great molecule, I think. It is a lucky charm. Why? Because you talked about only Fe2 plus and Fe3 plus. The Nobel Prize awardee, Professor Goodenough, he also got Nobel Prize only for LiFePO4 and for his lithium battery. There also he took only Fe2 plus Fe3 plus couple only. That couple is always giving a lot of Nobel Prizes, I believe. Even he never thought that he is going to do something for battery chemistry. He wanted to just study the thermocouple and the behavior only. But later on he arrived at as a great conclusion that the same Fe2 plus Fe3 plus couple could be used for battery chemistry, and this is how LiFePO4 came into existence. This is how he became the father of battery chemistry, and this is how he got Nobel Prize also. And next to him, now you are talking about this heme, and you made all of us to understand, even people like non-biology people like me, you made all of us to understand heme is a regulator, and your second molecule, curcumin, is a moderator. You clearly told it. And your heme regulator also, you made it in such a manner, if at all it is properly used, I think we can come up with very many discoveries. And curcumin, you generously made it in such a manner. Next to the mantra, water is an universal solvent. You said this is a universal adjunct molecule, or you called it as adjunct drug. So I think today I'm given to understand that the simple turmeric powder that we use in our kitchen, many a times things are very simple and very easily available, locally available. Very many times we happen to meet with them. We never even give them, we never respect them. We never even give them any kind of importance. But you made all of us to one more time understand that we need not worry about the Western people, their comments, their suggestions, and their beliefs. So for a country like India, it has taken for so many years. Now only we believed that the footwear that we are wearing is not suitable to us. We need not depend upon Western. We have to come up with our own Indian footwear sizing. Similarly, this turmeric science also will prove, and that will make Indians to feel proud in the global arena that whatever you said is a quotable quote. It is the universal adjunct drug. I think days are not so far off. Those who are working in biology, please target now curcumin, nano curcumin, so that you can also join his group of Laskar and Noble, so that the CSIR also will get some kind of recognition in the coming times. And sir, I'm very happy that you said that you also got a CSIR funded project, and you got one JRF and 5,000 rupees per month as the total grant for 1 year. That is one. And in one of your slides, just before the slide you showed, wherein you used piperin, you just, I forgot the name of the molecule there, within brackets you put CDRA. So I understood that either you or your students, they worked with CDRA, and maybe somewhere you put CDRA. And therefore, knowingly or unknowingly, already CSIR is there working with you, I think from 60s and 70s itself. And sir, before even I was born, you joined IISc, and now you are telling that I'm still going to the laboratory. And one particular line is very, very catchy, sir. You said that I am collaborating with, at the age of 86 plus, a leader is willing to collaborate with a company from Bombay. He is collaborating from Indian Institute of Science, he is collaborating with ILS, and he is collaborating with the Bombay-based company also. But we from CSIR at the age of 40, 50, or below 40, we always say that why only I should collaborate? No, I will work on my own. I know much better. My way of thinking is different. My thought process is different. Friends, it is not at all that way. See, the great leader at the age of 86, he clearly gives a statement that I am still collaborating. Sir, I'm getting a goosebump moment when I was just reading your statement. You are still collaborating and you are still chasing science, and therefore awards will continue to chase you, sir. With these few words, thank you so very much. I do not find the right word to appreciate you. And as I already told you, sir, all the 37 labs are listening to, they listen to your talk. They all will be excited to ask you many more questions, but they cannot ask because we've made it as one way. Sometimes we are so happy to make certain things one way. So only people here, they can ask questions. Maybe we can ask one or two quick questions. Before you start that, at the age of 86, 1 hour, he was able to stand and he was able to talk to us. It is not his physical strength. It is the mental strength and it is the strength given by science and technology. Thank you so very much.
Can I just, yeah. Yeah. So I would just like to share a few anecdotes. I don't know, Professor Padmanabhan remembers me. I am Geeta Vani. I used to work with Professor M.R.S. Rao as a PhD student. So I have known Professor Padmanabhan for 30 years, more than 30 years now, I think. He's popularly known as GP in IISc. We used to call him GP. And I saw him first when I was doing my MSc. He came to give a talk, and that's what inspired me. He and Professor Appaji Rao came. They were like rock stars to us. So the science, and it really inspired us to come. Yeah, even today they are, actually, you're right. So when I came to IISc, he was just across our lab. And in fact, M.R.S. Rao was his first PhD student of Professor Padmanabhan. And I just want to recall, he used to never walk on the stairs. He used to run on the stairs. I've always seen him running on the stairs. At that time also, he was almost close to 60, I think, probably. Even then, he was so active. And even on Saturdays, they used to call us for seminars, the molecular biology seminars we used to have, where Professor Padmanabhan and the whole department would come. And one thing which really, I think, 6 decades as you talked, many people started with classical biochemistry. In biochemistry department, we had professors who did only classical biochemistry. But what was unique about Professor Padmanabhan was he brought in molecular biology in the country. And I think that is really a hallmark, how he has adopted himself over the 6 decades. And then he contributed to the innovation ecosystem through BIRAC, and he talked of drug development. I knew him as a basic scientist, but today he is talking of molecules in clinical trials. I think that is a very unique feature, that he has transformed himself in 6 decades. And I think that's a motivation for all of us. Like DG said, he's only 86 plus, and I wish you many more success. And I just have one question to you. So you've done from 6 decades science. Was it easier to do science in 60s, or is it easier now?
It is difficult to answer your question.
Repeat your question.
So,
Ease of doing science.
No, I don't think that has changed. Attitude of people might have changed. You know, when I did science, I remember, as a student, we never asked when I will get my PhD. We never asked. Kept on working in the lab. But now the first question student asks is, when will I get my PhD? People have become very, what should I say, business minded.
Yeah.
This is how. But we were probably not very practical. I don't know. We just work. Just work.
one more thing I forgot to share. He's also a Carnatic music singer. Yes. He's very well known for his singing. And he used to always sing in the biochemistry department for many freshers party and all that.
Yeah, that's one solace for science. IISc of science. Can you repeat?
There's a YouTube video.
We have seen your song on a YouTube video. You're singing.
My singing?
In YouTube video. Yes, seen it.
I don't think so.
No.
Better not to listen.
Somebody has recorded in celebration of your award.
Can you say?
Sir, somebody has celebrated your award. At that time we were singing, so that video clip came at some place in YouTube.
I haven't heard my own singing. I can tell you that much. Whichever is the source.
Hello, sir.
A question we have.
Oh, sorry.
So because today I am not a biologist, but today I understood with your first work, because in our place there used to be this khesari dal. Khesari dal. Sir, I am Dr. Ramanoj right now at Bhubaneswar, but my birthplace is in Bihar. And Khesari dal, that what you referred in your first point, it was used very in general in 4-5 districts. And everybody was stopping people to don't take Khesari dal because your stomach upset will be there. Or if you are taking continuous, then you will get paralytic attack. And today I understood the science behind it, that why it is happening. And there was a band also that you should not use Khesari dal.
Yeah, but the dal I was talking is getting exported to Malaysia and other countries from India. So although there is a neurotoxin, I don't know whether it's responsible for the human disease. We could only show in the animal model. We haven't demonstrated in the human. We don't know.
So it was a poor man's dal at that time. In a dry land area, they used to give this dal.
Yeah.
So, sir, good afternoon. I'm Vibha, and you have chaired many of the project committees which I had coordinated. So, sir, in fact, my introduction to you was during my MSc because I also worked on Kesari dal, Lathyrus sativus, and on the toxin, right? So, for my MSc, of course, it was construction of the cDNA library, which was a first in IARI, where I was a student at that point in time. And then, of course, in my PhD, I had worked on the lectins of Lathyrus sativus. And the first, I think, two of your papers with Dr. Malati, I still remember that Malati and Padmanabhan, those were the two papers on Lathyrus sativus that were kind of, the introduction to us also on this particular molecule. So that was the first time I had heard about you and I'd read one of your papers. Then, of course, thereafter, during the meetings, I came to know not only the expert behind all this, but also the silent advisor, the person who was, even in the heat of all the arguments and all, who would remain silent and then ultimately, give his advice and his verdicts to all the members of the committee. So that is how I have known you. And just one question, sir. You spoke about two of the molecules, of course, Griseofulvin as well as Curcumin. So have you also worked on, Curcumin as an adjuvant to Griseofulvin and seen whether that, that entire compound is more impactful than the artemisinin plus curcumin molecule?
No, my model has been only cerebral malaria.
Okay.
But the point I was trying to make is we should not dismiss. There is a tendency to say these are all described in your old literature, they are not true. That's not correct.
Yeah.
I was trying to make, there is a science behind. I don't know how they knew the science, but there is a science behind. Today we understand how curcumin acts, probably a small aspect of it. It's an adjuvant. Once it's an adjuvant, it will work with almost any protein.
Okay.
Therefore, it doesn't have to be protein specific, it doesn't have to be disease specific. It will work on every molecule. It's an immunomodulator. This is basically what.
And piperin will probably increase its bioavailability in the body?
Antibody?
Piperin will probably increase its bioavailability.
No, piperin is an inhibitor of phase 2 drug metabolism.
Yeah.
So it inhibits the metabolism of curcumin in brain, in the blood.
Okay.
Therefore, the half-life of curcumin increases. So one of the criticism has been that curcumin is eliminated very quickly. So if you make a combination of curcumin and piperin, its half-life increases in blood.
Okay.
That was the.
Thank you.
Good evening, sir. Myself, Dr. Meenakshi Singh. Uh, I was coordinating several projects and you are chaired under Nimitli scheme. And my question is, of course, you have touched very important points like Lathyrus sativus and curcumin. Curcumin now, many people, you also said that nano curcumin, one word. And many people now want to use it because as such curcumin is not that much bioavailable and they want to make it more bioavailable. That's why they want to make it nano curcumin. But there are several aspects of safety related issues which have been not proven right now, and that's why we are not able to use nano curcumin. So some light if you want to throw
I
it.
don't know. This is where the problem comes, in my opinion.
Yes, sir.
Many of them dismiss nanocurcumin because of safety aspect.
Yes,
What
sir.
safety aspect? That nobody tells me. What is the safety aspect we are worried about? Will curcumin become toxic now?
Yes, sir.
Why should it?
Because
No,
there are several, I think, studies. IIT Bombay also they have.
no, nanocurcumin may be more absorption. Takes care of the issue.
Yes.
Curcumin is poorly absorbed. Curcumin is metabolized faster. So you use nanocurcumin. Now, that was the reason why nanocurcumin was suggested actually. But it's been made now. A lot of groups have made nanocurcumin.
And of course, it is.
Internationally, they have made nanocurcumin. My plea is, it's a molecule which has very wide application because it's an immunomodulator. It's not acting against one disease. It's not acting against one target. That is where its mechanism is different.
More question.
Yeah. No issue. No issue.
Sir, I'm not a science student. I'm at the, I will say, middle age. I'm just curious to know, sir, that among your three innings you mentioned first, second, and third, which one you enjoyed the most?
You have to repeat for me.
I mean, I'm at the middle age, I will say, and I'm just curious to know that among your three innings, first inning, second inning, and third inning, which one you enjoyed the most? Just out of curiosity, I'm asking, if possible.
Second inning, third innings. Which one do you like? She asked.
Which one?
Third innings. First inning, second inning, third innings. Which one do you like?
No, I only think I will go as long as it lasts. I don't plan much. I'm prepared to take life as it comes. But, I don't give up like that. So people talk to me in Bangalore itself, A lot of people want me to come visit their lab. Many times I go learn. I don't know the science. I don't know the technology. But it gives me an opportunity. If, you feel wanted. This man, an old man, is retired. That doesn't give you that kind of feeling. So you are still useful to the society. Continue as long as it lasts.
Thank you.